All right, everybody. Let's see here. Expand this out. There. We still have, I guess we still have a minute or two um, before official start time. So I'll take my time, and then I'll explain kind of what we're going to do tonight and why it's a little bit different. First of all, let me ask you: does my microphone does it sound normal? Or do I sound like a cartoon character? That's what someone said earlier. Does that sound normal?
Sounds normal to me.
Okay, good. Um, I guess I had a problem or something earlier in the day during office hours, so just making sure that it's um, not torturing you. So I think, yeah, I think that's good. So what we're going to do tonight, and so you might notice that maybe not everyone will show up tonight, um, and that's what happened in my afternoon class. Um, to make a long story short, um, we have a Wednesday holiday. Cesar Chavez Day's on Wednesday this week, and so so for the Monday classes, you guys Monday class, um, we have the normal Monday class, office hours, and then the two labs. But um, the problem becomes then on Wednesday, the people that have Wednesday labs, um, they still need to get together. They still need to do the lab, one way or another, and they need to get together. In particular, this lab, because you have to uh, talk about your germination experiment. So this was um, a little bit of an issue with the holiday that no one saw coming until I kind of realized it on Friday. I'm like, hey, we're going to have a little bit of an issue here. So what I decided to do, and I've done this before, and I think it works pretty well for the most part, but to keep it sort of fair and keep everybody on the same page, um, Monday today, I'm teaching my regular class because I'm supposed to, because there's no holiday today. I guess I could do it fair and I could just not show up Monday either. Should not show up Monday and Wednesday. Then it'd be fair. But so what I'm going to do is I'm going to do my regular Monday class, and I did this morning as well. And I'm going to record it, which I'm doing now. And then I'm going to edit the recording so that I'm the only one that you can see in the video. So by the time we're all done today, I will make a copy of this lab and I will post it on my website for students that couldn't make it to class. They can do the lab asynchronously. But again, there's nothing you have to do on your part. You won't be in the video because I can edit you out, which I've done before, so that's no big deal. Um, but to keep it fair, I also invited some of the Wednesday students. I told everybody in my email that if you have lab on Wednesday and you want to join us on Monday, you're welcome to do that. And what you would hope would happen, which didn't, is that everybody saw the wonderful benefit of meeting directly with me so that all the Wednesday students showed up at 1:15 and at 7, so that we had actually 48 people in each class. But as you can see, we don't. But that's sort of what ends up happening. So personally, if it were me, I would suggest that the regular synchronous class like you're doing now is the better way to do it, because as we do it, you can ask questions, and particularly in class, I can explain when um, when we hit something that maybe you don't understand. I can kind of tell, oh, I don't think they're getting this part. So personally, I think you learn more when we have it in that synchronous mode. But because of the Wednesday holiday, I'll record this. I'll make it available. And if you want, if you missed something today, you could go back and watch it. What a lot of people are doing, particularly in the beginning of the pandemic, is and I just had a meeting with my boss about this, but they would essentially do it one time, record it, and then just play it over and over again for all the other classes, which is a very fast way to get things done, but it's not very conducive to, I don't think, how you would learn it. So when I'm done recording this part, then I've got to go back and edit it after we're done to take out the pauses and all the various things of that sort. So it will be, I'll post it up here, and you'll be able to find that if you want to review Lab 6 as an asynchronous lab. But since you're here, you won't have to do that, most likely, but it's up to you. Couple things real quick. You'll see that, first of all, there's 2 worksheets, and you're going to want the second one that's an updated version. So when you're working on it today, you're going to want this second one. And then you can record it. You can, after you're done, you can save it in that same spot. The other thing I did was, in the Lab 6 module, I put a discussion board that I'm calling the Radiseed Germination Experiment Data, and you'll see that, and no one responded yet, which is kind of good, but for the Monday night people, and there'll be one for Wednesday afternoon and night also. There are 4 groups, and you might remember that last week you met in a group, designed your experiment, and you collected your data. But what group number you were in is irrelevant, right? So here I call them group 1, 2, 3, and 4. The number doesn't really matter. So what you're going to do is, when you have your data, go ahead and pick any one of these spots. Like, oh, you know what? Group 2. No one's in group 2. If none of the people in my group have said anything, I'm going to be the first one. You put your name in there, your experiment, and then you put your results in there on how many germinated or not. And hopefully then everybody in your group will go, oh, they were in my group, so I'm now in group 2. And they put their information in group 2. Does that make sense? So the actual number doesn't matter per se. It's, if you put your name in there in your data, and then after that we'll just follow whoever sort of took the first initiative. Okay. Also, what we'll find is that this kind of thing where you kind of open it up and let people do things kind of when they want is not always very successful from them. So what I suggest you do is you put your data up today or tomorrow, and then by like Thursday, let's say, if your group members didn't put your data up or their data up, don't worry about it. Take the data that's there and make it work. Don't agonize over the fact that your group members didn't do their part. Just move on. Give them a chance to Thursday or so, but after that, go ahead, just use what you've got. Does that make sense? That way you're not sitting there trying to email them and freaking out about that. Just forget it. Okay? At that point. Hopefully everybody does, but when people kind of have it opened up, they do different things. All right, so I've got that. And once again, just like we had before, we had our quiz today. In the first class, I didn't see any problems, but if there was a problem with the quiz, then once again, you have, not there, did I? Oh, I have it twice. That's why I couldn't find that. Did you guys put it there? I ended up putting it in there twice. So since no one has used this one, I thought I had done it and I couldn't. I'm going to delete this one. So we'll leave it in with the lab. So there we go. Yes. There we go. Okay. So after you take the quiz, anything wrong with it, you can put it in the injustices. As I said in my email, though, I'm going to, because of the Wednesday class, I'm just going to open the quiz up, and you can take it anytime between now and Friday. Um, and you can also turn in the lab that technically is due today. You can turn it in by Friday. However, keep in mind, this is the downside. I'm giving you more time to do it because of the Wednesday class, making it a little more flexible. But where you get burned is if you kind of slack off till Friday night, and you're like studying and you take the quiz and then you turn your lab in. Well, then the next one's due Monday. Like next week there's another quiz and a lab due. And if you're like, oh my gosh, I got 2 due in 2 days, well, that's because you waited. So, that's your fault. So just make sure if you procrastinate, it gets tough when you come back that following week. Okay? So let's see. I think that's all for the intro stuff that I needed to do there. Does anybody have any questions before I start this part here? Nobody? Okay. Where's my pointer? There we go. All right. So, oh, you didn't like that. Okay, let's do, here we go. So hopefully, as I mentioned last time, we did cellular respiration. What we're going to do today, photosynthesis. If you kind of don't really understand cell respiration, if you kind of just kind of get it, then the problem is photosynthesis is going to look very, very similar. So when we do these biochemical pathways, it's important to have one down pretty good before you try to learn the next one. And so we'll start just briefly by talking about, remember when we did cell respiration last time, there's a couple of things that happened. One is that we used up sugar and we used up oxygen, and some of the products that came out of that were water and CO2 and ATP. And that, of course, was the main goal of cell respiration, is to make ATP. That's what we're trying to do. Today what we're going to talk about is photosynthesis, and one of the things you'll see really quick that's kind of interesting is that the inputs of one are kind of the outputs of another, and vice versa. So photosynthesis, for example, well, let's start with the inputs here again. So cell respiration, if our inputs are oxygen and glucose, what we're getting out of that is CO2 and water. In photosynthesis, the CO2 and the water are inputs coming in, and they allow me to give off oxygen and sugar. So they're kind of opposites of one another, and this is not just by coincidence. One of the things that people miss a lot, so I'm going to try to make sure to tell you very carefully here, is that when we do this, you get the idea that you and animals do cell respiration. It's got the mitochondria. Mitochondria are in animals. They do cell respiration. Perfect. Okay. Plants. Okay. Plants are green. They have chloroplasts. Chloroplasts, they do photosynthesis. Okay. However, what people miss a lot is you have to realize that plants are actually doing both. So plants do photosynthesis to make the glucose, but then they do cell respiration just like we do to make the ATP. So plants are doing both photosynthesis and cell respiration, not just one of the two. That's a very common thing that students get stuck on, is they start to assume the animals do one and plants must do the other. And that's not quite true. Okay, so the plants are doing both of them. In fact, the plants, in part, the plants are really doing photosynthesis in order to make the sugar so that they can then produce ATP. So for the plant to grow and do things like other living things, it still needs energy, and it's not really getting its energy just from the sun. That's just one of the steps. It's going about that to eventually end up getting the glucose to go through cell respiration. Okay, so that's what's happening there. Photosynthesis will have 2 stages, kind of like when we did the 4 stages of cell respiration. And if we take something like a leaf, where a lot of the photosynthesis takes place, inside the cells of the leaf are these structures here, and these are the chloroplasts, and the chloroplasts are the organelle where the photosynthesis takes place. And just like the mitochondria, the chloroplast has multiple layers. So there's an outer layer, and then there's a couple inner layers. So like having the multiple layers in the mitochondria, you have sort of the same setup in the chloroplast. Okay. Now we have, like I said, 2 sets of reactions, and they call them different things depending on where you look. Today in lab, we're going to call them the light-dependent reactions and the Calvin cycle. Sometimes in older textbooks, you'll see the dark reactions or the light-independent reactions. So there's various ways they describe the second part. They call the light-dependent reactions sometimes just the light reactions. But the idea is that the light reactions, they require light. And part of the problem with the dark reactions is people started thinking, when you would teach it, that the dark reactions must require dark. And that wasn't true. They just don't need the light. So the light independent reaction is probably a better name. Also, it was discovered by this guy named Calvin. And so some people call it the Calvin cycle. So we'll talk about that too. Okay? Now, the first thing is when we talk about the light here, light comes in. If we talk about normal, what we call white light. White light is essentially all the colors, and sunlight and many white colored lights have these multiple colored wavelengths in them, which you can't see unless you separate them. Okay, so it turns out that something like a leaf, if a leaf is green or brown or whatever it is, the color that it is because it reflects that color of light. So leaves are green because when the white light hits them, the green color bounces off the leaf and it travels back to your eye and you perceive the green color. So the plant itself is, like everything else, is made up of these atoms that have electrons spinning around them. It's mostly empty space. So when you perceive a color, it's because of how the light hit that particular object and bounced back to your eye. So the green that you see is because the green wavelength bounces off. And then if we were to measure, there's certain devices that we use on campus that measure the absorption index, what colors are absorbed or wavelengths are absorbed by certain molecules. And what you find is that there are kind of 2 peaks, and one of them is a red peak and one is a blue peak. But these are the 2 colors that correspond mostly to what is actually used by photosynthesis. So plants reflect green light, but generally absorb red and blue light, and that's what we're using in photosynthesis. Okay? Now, this might look familiar to you because it should look kind of like the electron transport chain in cell respiration. But this one's in photosynthesis. It's still an electron transport chain, but instead of this being powered before, like when we had NADH, right? Instead now, where that high energy push is coming from to push an electron to a higher energy level, instead this protein is reactive to light. So it is light that allows me to generate that high energy electron. And the way I get the electron is by a reaction called water splitting. So water which we made last time, remember when we picked up the electrons in cell respiration, has an oxygen and two hydrogens. And if you split that, you can pull the electrons off, and that results in giving off O2. So the reason plants give off oxygen is it's a byproduct, it's a waste product of the water splitting they do right here to get the electron. Then, just like before, we have this electron transport chain, and we push that all the way down to make a hydrogen ion waterfall kind of thing again, like we did before. You'll notice though that instead of NAD and NADH, like we did with cell respiration, instead we have a very similar molecule, but we end up making NADPH. So that is kind of unique to photosynthesis. So instead of NADH, you have NADPH. Again, cell respiration, if you kind of like, yeah, I kind of get it, well, then you're like, this looks exactly the same. It's not exactly the same, but there's a significant amount of similarity that if you don't have cell respiration down, this looks similar. So you kind of have to, make sure you got that down. Now, eventually what will happen is I'll generate some ATP doing this, just like I did before with my ATP synthase. But the goal of the light reactions really, making ATP is useful, but that's not really what I'm going for. What I'm really trying to get is this NADPH. This is the significant part right here. Okay, I'll point out here that you'll notice that this is photosystem 2 and photosystem 1. Why the hell would you have photosystem 2 first and then photosystem 1? They're this way because that's how they were discovered. They discovered this one first, and then we were trying to figure out where the electrons came from, and later on they figured out that's photosystem 2. So even though it goes 2 and then 1, in reality the flow of it would start with 2 and go to 1, but they're named after when they were discovered. Okay, and then those are light reactions. Remember, trying to make NADPH. And then what we do in the Calvin cycle, or the dark reactions, is we're going to take this enzyme here called Rubisco, and Rubisco, along with my NADPH and some of my ATP I've built, I'm going to take in carbon dioxide, and my Rubisco enzyme, along with ATP and NADPH, allows me to make this G3P, which, if you go back and look at all the steps of glycolysis, G3P is one of these intermediates to glucose. So, in a nutshell, if you can make G3P, you can make glucose. So the whole point of photosynthesis is to make glucose, and the reason the plant's making glucose is then it can use the glucose to go through cell respiration to make ATP. Okay? All right. Now, when we talk about plants, most of the photosynthesis that we're talking about, of course, is going to take place in the leaves. And we're going to do a couple of generalized plant sort of parts. We're going to talk about some plant parts today and what those parts do. And in doing so, I'm going to keep it general. And so the first thing I'm going to tell you is that we're going to take a plant and sort of describe it based on two parts. The part that we typically think of as being in the ground, the root system, and the part that is above the ground, which is the shoot system. In reality, just so you know, there are plants, of course, that live on the sides of trees. Their roots aren't in the ground, epiphytes and things of that sort. So there are a lot of exceptions to this. There's specialized roots and specialized stems that do a variety of things. Most plants, though, that you're familiar with fit this sort of model. So we'll stick with that just so you know. Okay? And so everything above the ground is the shoot system. That includes the flowers, which are used for reproduction. We'll talk about that. And the leaf, which is primarily used for photosynthesis. And then everything below the ground, again, we have the roots. And the roots are mainly used for absorbing water and minerals. And they also help hold the plant in the ground. So they anchor the plant to the ground so it doesn't blow away and things of that sort. Okay? Now, when a plant gets larger, so we have our bean plant and we have our radish seeds, which are very, very small at this point. But as a plant gets much, much larger, there becomes more difficulty in transporting the nutrients throughout the plant. So one of the things you'll see as plants get larger, especially like the size of a tree, is that we'll see larger and more collective groupings of these two specialized tissues here called xylem and phloem. And xylem is a tissue that is used by plants to transport water. It's found in the roots, but it's found throughout the plant as well. And then the phloem is also a specialized set of tissues and tubes that are designed to transport food. And food usually is in the form of sugar. So kind of as a good starting point, you can think of it like this. In a big tree, the roots are where the water is. So getting water into the roots is not a big deal. But in order to get it to the top of the tree, you need the xylem to have a way to have a tube to get it up there, right? Why do you need water at the top of the tree? Well, if the tree is going to do photosynthesis, remember water is that input for the water splitting. So you need that. To do photosystem 2, you've got to have water. So you've got to get water from the roots all the way to the top of the tree. Likewise, the roots, they're underground. They're not doing photosynthesis usually. So if they're going to grow or absorb water or do whatever activity they've got to do, they have to have energy. Well, where are you producing the food? You're producing it usually in the leaves. So you got to get water in general up the tree, but you got to transport the sugars down the tree to get them, kind of equally in most parts. Okay. So the first activity that we're going to have you do tonight, I'm going to go through them and then I'll put you in breakout groups and you can start on them. The first one we're going to have you do is we're going to have you look at root hairs. And we're going to have you use your your radish seeds. So hopefully at this point you have a good radish seed. And if you take one of your radish seeds and it's germinated, first of all, make sure before you go using up all your radish seeds, make sure you get your data on which one's germinated and that sort of thing. But you're going to take one of your roots from your radish seeds and you're just going to cut off a little tiny piece. Okay? And when you look at this, when you take your radish seed and you cut it and you just look at it, you'll probably see something that looks like this. It's a little tiny root. Okay? Little tiny, thin, white colored root. But then you're going to take that, put it on a microscope slide, put a drop of water on it, and put it under your microscope. And what you'll see is that one thin little piece is going to have on it, and you might be able to see this without the microscope. I don't think I can. But you'll see it has these little tiny things sticking off of it that are usually, they're microscopic, usually you can barely see them. You can kind of see them with your naked eye, but it's much better under the microscope. And that's what you're looking for there. Those are the root hairs. And then what you got to figure out is, what's the importance of those. And we'll go back at the end. We'll talk about that. Okay. The next activity that you can do for this is you get a piece of celery. You cut the bottom of the celery off. The celery, the part you eat usually is the petiole, and then up at the top here is the leaf. So this is like the stalk or the stem of the celery. So you're going to cut it, and then you're going to put it inside a cup with food coloring. And it doesn't matter what color food coloring. Any food coloring you got, put a few drops of water in the food coloring, stir it up, cut your celery so you have a fresh cut, put it in there. It takes around 30 or 40 minutes. And then when you're done, you'll look at that, you'll take a picture of that, and you will try to figure out what that is there. And I'll give you a bit of a clue. If you were to look kind of at a bigger scale of those under a microscope, you might see this combination of tissues together that group together. We call it a vascular bundle. And within that vascular bundle, you will often find both phloem and xylem together. And remember, the phloem is used to transport food, and the xylem's used to transport water. So what you see as a tree gets bigger, as a plant gets bigger, the ability or the need to transport more water also increases. A little tiny plant can transport water no problem. But as a plant gets much, much larger, its demands to transport food and water also get bigger. So a lot of plants that are really big, primarily certain kinds of trees, will form what we call secondary xylem. And these here are the layers of secondary xylem, right? So you've got one layer here, you've got one layer here, and you've got one right there. And they grow from this point right here. So if this is a tree, if I've cut a tree in half, and it didn't come out very well. This is just a quarter of that circle. And so this is the outside of the tree. This is the center of the tree. And it grows from this spot right here, this section right here. And it actually grows both ways. It grows this way and it grows this way. This stuff we rarely talk about. It's called secondary phloem. It does the same thing as the secondary xylem, but for food. But it's usually compact and you can't really see it. So the only thing we usually kind of look at is this secondary xylem. And what happens is this ends up being very useful when you're looking back at climate data. It's one of the ways we can look at climate data because we know that these trees form these rings every year. And so what you can do, if you count, you can count how many years old this is. And what you find is like this year, right about these 2 years here, for example, or these 3 years, let's say, these 3 years had more rain than these years because the trees, when they have more rain, they grow faster, they grow bigger rings. And when there's less rain, they have smaller rings within the same kind of tree. So you can't compare like a walnut to a oak tree necessarily. You got to have the same species of tree in your comparison. And many of these go way back. So like this one here, this tree, this goes back to 1637. And we have some of these tree rings in California, actually in the Eastern Sierras, that are thousands of years old. And they can do these, there's an instrument called a boring tool, and you can stick this in the tree, you can pull out this segment, and you can count these tree rings and how old it was and when certain things happened and that sort. Some of these go back thousands of years. And just in case you're wondering, the idea of the cork borer is that it doesn't kill the tree. So, you know, we're not just killing a 2,000-year-old tree down to count the rings. If you do it correctly, the tree lives everything goes on fine. There are some of these that are thousands of years old. These bristlecone pines, they're thousands of years old, and researchers that study them, they don't even let you know where they're at. You know, like they're not. There's no signs. They go, this is the oldest tree, because they're worried that people get, drunk and go cut them down and stuff like that. Because that's what would happen with some people. Like, I know what's funny. Let's go cut down a 3,000-year-old tree that's made it all the way until this point in time. So anyway, a lot of them keep those trees secret. The next thing we're going to have you do is going to involve your bean plant. So hopefully your bean plant has some leaves on it at this point. And what we're going to do is we're going to look for these structures called stomata. And you're going to pull a leaf off your bean plant, and you want to look at the underside of the bean plant. So you got your bean plant. When you're ready, you pull the leaf off, and the part that would hit the sun is the top of the bean plant, and the bottom is the underside, which makes sense. You're like, of course, I get it. But what some of you will do is you'll pull the bean plant leaf off, and you'll go put it down on your table, and then you won't remember which one's up or down. So make sure that when you look at it, you're looking at the bottom side. That's the goal. And again, the thinner the better, bottom side. And what you're looking for is these two things, really. And these will be very small. They look like, they look kind of like, I always think they look kind of like lips. They look kind of like, I've heard them described that way. Like they look like little lips. I'm having trouble with that part of the lip. That doesn't look very lip-like, does it? Okay, let's try. Here we go. There we go. That's a little bit better. Anyway, the hole there, that hole there, that's what you're looking for. Those are called the stomata. And then around it, what surrounds the hole, the lip cells, those are called guard cells. And what the guard cells do is they can, in many plants, they can open and close, and they let in the carbon dioxide. That's how the plant does the Calvin cycle. But the problem with them is that when they're open and the carbon dioxide can come in, a lot of plants lose water through the stomata. So plants often have a way to close the stomata if they run out of water. So that's one of the problems that plants have, is that they tend to they have to open the stomata to get the CO2, but if it's too hot out and the soil's too dry, they end up losing too much water and they'll shut down. Okay, so there's that. Then we're also going to look a little bit at the seed structure. We already kind of, you don't need to put this under your microscope. We took our radish seed, and just so you have an idea of what we did, we took our radish seed or our bean plant, and what the seed is sometimes what we call it's basically a baby plant in a package. So a seed is it's got a plant in it that can grow, but it's packed also with nutrients, because when you first start with a bean or a seed, what has to happen is there needs to be a certain amount of energy to get the plant started. So the seed has stored up energy in it so that the plant can get started. And once it grows its first leaves, its cotyledons, it can start doing photosynthesis and start doing what it needs to do. So plants need this sort of boost in the beginning, even though they make their own food. In the beginning, they're kind of stuck without it. They don't have anything green yet. So that the nutrients stored up in the food allows them to do that. And then the last thing, and I'm going to suggest in this case, if it's dark outside, it looks like it's dark outside right now, you might want to do this during the day, but we're going to have you go out and try to find a flower, and then there are apps that you can get on your phone. There are different ways you can do this too, but probably these days the easiest way for many of you will be to download an app like iNaturalist or Seek, and with flowers at least, you can walk up to the plant, you can take a picture of it, it sort of scans the plant, it asks where you're at, and it kind of tells you what it is. So it's kind of a quick, easy way to learn to identify plants these days. If you have books and you do it that way, you can do it as well. But what we really want you to do is to find a plant, and what you'll find most likely is that you'll find a plant that has flowers on it that are what we call a complete flower or a perfect flower. And what that means is it has both male parts and female parts on it. So most flowers, most plants that you would know, have flowers that on the same flower have both male parts and female parts on the same flower. The male parts are called the stamen collectively, the female parts are called the carpel. And that's the most common kind of flower. However, there are flowers where there are just male flowers, and then separate from the male flowers there are female flowers on the same plant. So you can have a plant, certain plants have on the same plant will have male flowers on them, and then elsewhere on the plant will have female flowers on them.